So, hello everyone. My title of this talk is Role of Friction in Ciliary Beating and Phototaxis. So, this work has been funded by India Alliance DBT Welcome and my host institution, Indian Institute of Science. So, before I tell you about the crucial role of friction in ciliary beating and phototaxis, I would quickly acknowledge the people who have been involved in this work. So, the ciliary beating work has been done by my graduate student Devaswita Mondal. We worked in collaboration with Professor Rona Jayadekari. And the work on phototaxis has been done by another student, Sujit Kumar Chaudhary, and we worked in collaboration with Professor Prerna. So let's switch to the real title, which is to probe roles of friction in these two important phenomena. So the outline of this talk is that I'm going to tell you about what active filaments are, and cilia are essentially a class of, example of active filaments. And then I'll discuss some of the key experiments that probe the role of hydrodynamics in ciliary beating. And then I will also highlight some experiments that have attempted to measure internal friction in passive biopolymers. And after setting all this background, I will present to you the measurements that we have done on an isolated cilium. Specifically, we have looked at measuring its waveform and flow field. And that will be the part one of the talk. And the second part, I will talk to you about phototaxis of single-celled algae. And phototaxis is essentially a stimulus response kind of behavior where the cells run towards a source of light. And this directed motion is controlled by ciliary beating in the sense that in pitch dark, the cells would be swimming in random directions. And then as soon as the light is switched on, the ciliary beating is controlled in such a way that the cells turn towards the source of light. And then I will conclude. So active filaments are non-equilibrium chain-like systems that undergo spontaneous oscillations by converting chemical or other forms of energy into mechanical motion. So first I'll show you some examples of synthetic active filaments. And so here you're looking at, let me switch to a pointer. Here you're looking at a chain of paramagnetic colloidal particles that are bonded to one another through a polymeric glue. And you can actuate this filament by applying time varying magnetic fields. You can get really exotic kinds of motion if you use Janus particles. So Janus colloidal particles are essentially half dielectric and half metallic. So their surface is heterogeneous. All these phenomena are happening at the micron scale. And essentially these particles are placed in a salt solution, and a AC field, electric field is applied perpendicular to the imaging plane. So the attractive interactions drive the chain formation, but the motion of the chain comes about due to induced electroosmotic flows. So I'll play the movie. Essentially what's happening is that in presence of the electric field, the two sides of the spheres are polarized differentially. So the metallic side is much more strongly polarized than the dielectric side. And this drives induced electroosmotic flows, which are asymmetric in nature. That means the metallic side is facing locally more flow than the dielectric side. And that causes this chain to, each particle of the chain to propel. There have been theoretical studies where the authors considered a chain of active colloids, polar active colloids, which essentially underwent self-induced buckling instability due to the compressive stresses of the the active spheres, imposed by the active spheres. So all these examples are trying to mimic what is called ciliary beating. So cilia is a key organelle in biology for which is used to set up fluid flow and transport. So this is how our lungs look like. So you're looking at an epithelial tissue, epithelial layer of the lungs, and these hair-like structures that are faintly visible right now are cilia, and their function is to basically beat in the fluid and set up fluid flows. So I'll play the movie, and you can see that the filament are going back and forth, and this back and forth motion sets up fluid flow. And it's really important for our survival because this fluid flow pushes the mucus and the debris out of our lungs. So if you put tracer particles on top of this of this tissue, you can visualize the unidirectional flow. So here we zoom out, and you can visualize how these tracer particles are moving unidirectionally. So essentially, mucus clearance in our bodies comes through coordinated activity of thousands of multiciliated cells, each containing hundreds of cilia. So such macro scale transport is essentially coming out of beating of thousands of cilia at micron scale. So the interesting thing is that ciliary beating is not just important to higher order organisms like us, it's equally important at the micron scale, or for that matter, microorganisms. So here you're looking at a 50 micron paramecium cell whose surface is covered with cilia. So what is happening is that these cilia beat and they push the fluid, and the fluid pushes back on the organism and propels it forward. So I'll play the movie. What you will also see is that there is beautiful coordinated motion of cilia across the cell body. You can see long wavelength modulation of the phase of the beating across the carpet. These waves of phase are also called metachronal waves, which the next speaker, Professor Benjamin Friedrich, will discuss in detail. So far I've shown you examples where hundreds of thousands of cilia are involved. And you may wonder whether even one cilium, how good it is. In fact, it is extremely crucial for our life, our existence, because the sperm swim through the reproductive tract through the motion of a single cilium. So I'll play the movie here, and you can see that this single cilium motion is good enough to propel the small sperm forward. Another model organism that uses few cilia is Chlamydomonas reinhardti. It's a single-celled algae, about 10 microns in diameter, and it bears two flagella or cilia. So most of my talk will actually refer to this organism, both in terms of background experiments and in terms of the actual work. The reason being that this is a model organism which is easy to grow and easy to maintain. You can get a large number of mutants from worldwide repositories. So you can see how the cilia on this cell beat. This cell has been held on a micropipette. If it were not held on a micropipette, it will actually run away. And so you can see how the two cilia are beating in a very coordinated, synchronized manner in what we call a breaststroke waveform. So the physics of synchronization will also be discussed in detail by Professor Benjamin, and I will refer to some of those experiments as well. Now, the interesting and the helpful thing is that the microstructure of the cilia is nearly identical between human beings and Chlamydomonas. So it's evolutionary conserved, meaning right from single-celled organisms to higher-order organisms, the microstructure of cilium is nearly the same. And the core structure of the cilia is what is called a 9+2 microtubule doublets. So microtubules are these rigid rods shown in blue here, and they form a ring-like structure surrounding a pair of microtubules at the center. These microtubules are also anchored to one another through molecular motors, which are dynein in this case, and there are hundreds of dyneins all across, all along the length of the filament. In fact, they are repeated, they're present in a repeated periodicity across the filament. And the whole structure is encased in a plasma membrane, which is shown in here in yellow. So how does this structure of motor filament assembly produce this kind of motion? So what is going on at the micro scale? So at the micro scale, what is happening is that the dynein motors, their role is to slide microtubule doublets. So the motors consume ATP, which is the source of energy, and apply these shear forces on the microtubule doublets. These doublets are actually cross-linked to one another through passive proteins like nexin links, and these internal cross-links convert the sliding motion of the doublets into bending. This whole organelle is working at what we call low Reynolds number. So the length of the filament in the case of Chlamydomonas is about 10 microns, and the diameter is about 200 nanometers. The wild type peak frequency is close to 60 hertz. So essentially, if you calculate the Reynolds number, it will turn out to be 2 orders of magnitude less than 1. In such a scenario, you expect the inertia of the system to be irrelevant and friction to dominate. So one of the most obvious sources of friction here is the surrounding fluid. The key concept here to remember is that this is a continuously driven system. The motors are continuously consuming ATP and doing work. And so if you want the filament to beat in a steady state manner, then that can happen only when the elastic stresses due to the drive, which is due to the motors essentially, are balanced by significant dissipation. And this is the source of dissipation is what has gathered recent attention of the scientific community, which I will come to in due course. So traditionally, ciliary beating analysis has been done with fluid friction alone as the sole source of friction. And what is usually done is that you measure the waveform of this beating cilium, and then you write down equations of motion that balance the active moment due to the motors and the passive stresses. So what are the passive stresses? The passive stresses are the elastic energy or the elastic forces to bend this filament and the fluid friction. And the active moments are due to the molecular motors. Now, the subtle point here is that a priori it's not known what is the constitutive equation for the active moment. By constitutive equation, what I mean is how are the active stresses related to the observable variables in the experiment, like the shear angle or the sliding displacement. So essentially you try many different models of active moment and solve for waveform, optimize parameters to get a best fit. So for example, as shown here, so you're plotting the fundamental mode of the tangent angle at each point, and then comparing it with the theoretical prediction. So notice this is an inverse problem that you write down equations of motion and then you are trying to estimate parameters that will lead to best fit for the experimental waveform. So this is the background of, or a short background of ciliary beating. And as I mentioned that the source of friction has been something which has been debated in the scientific literature in the context of ciliary beating. And a lot of literature has focused on obviously probing the role of hydrodynamics, and the way it has been looked at in the context of chlamydomonas is to look at in the context of synchronization. So the idea is that the fact that the two cilia on chlamydomonas are beating in a synchronized manner, then they must be coupled to one another through the surrounding fluid. So, to probe the strength and the nature of this hydrodynamic coupling, a number of experiments have been done, and I'm highlighting a few of them here. So, in this experiment, the authors held the cell on a micropipette. Essentially, it's like a glass needle on which you hold it, and then you apply periodic flows, and then you ask for what parameters of the periodic flow will there be phase locking between the external flow and the flagellar beating. So what these authors figured out was that yes, for certain parameter range you do find synchronization, but the magnitude of the hydrodynamic forces that were required for this kind of synchronization were greatly in excess of what the cell would encounter in its physiological settings. And so they concluded that there must be a synchronization mechanism that is internal to the cells, and that was ascribed to what is called distal fibers. So the two cilia that are present on the cell body are connected to one another within the cell body through these elastic fibers called distal fibers. And so essentially there is some kind of internal elastic coupling between the two filaments that could in principle lead to synchronization. And it was also said that this was an actively regulated mechanism because the strength of the distal fibers could be controlled through intracellular calcium level control. So essentially, the conclusion of this these set of experiments was that hydrodynamic synchronization was probably not important in the context of biflagellate algae, and there is some cell internal mechanism. Another landmark experiment, in which Professor Benjamin was also involved, was to look at flagellar beating in presence of steady flow. So instead of applying periodic flows, you apply steady flow, and you measure the changes in the flagellar beating, in particular changes in the amplitude and changes in the phase. And you can plot the amplitude and phase susceptibility and write down equations of motion to figure out whether such a behavior can be explained. And what was found out was that even to get a qualitative fit to the experimental data, an extra source of friction was required. So standard fluid drag was not enough to capture the changes in flagellar beat in response to a steady uniform flow. And what the authors did was to model internal friction as some kind of effectively renormalized fluid friction, and found that the theory and experiment match can be made. So in this experiment, there is room for improvement, in the sense that currently the internal friction is just the same as fluid friction, and with a magnitude which is scaled up. And also the fact that all of these experiments so far, not just this one, but the ones that I had mentioned before, were conducted on live cells. And essentially, live cells are subject to intrinsic biological variation. And also the fact that the cell itself, the cell body or machinery itself, imposes control on ciliary beating. So what we wanted to do was to study the mechanochemistry of motors alone. In a sense, we wanted to look at how this motor filament assembly behaves regardless of the regardless of the other biochemical pathways that are taking place inside the cell. So now I will show you experiments which looked at existence of internal friction in passive biopolymers. And these are generic biopolymers that are also relevant to ciliary beating. So I've shown you experiments which were looking at role of hydrodynamics, and now I'm going to show you experiments which are going to assess if there are internal friction mechanisms present in biopolymers. And so in this study, the authors looked at the relaxation timescales of the bending modes or bending fluctuations as a function of the wave vector for two classic biopolymers, microtubules and actin. So microtubules are the building block of cilia. So if you write down simple equations of motion, here you're writing down a force balance kind of equation. So the first term is essentially the curvature elasticity. The second term is the viscous force, and the third, the term on the right hand side is the thermal noise. So U is the transverse displacement of the film. So from here you can get a time scale which scales as 1 over q to the power 4, which works well for the actin case, but it doesn't work well for microtubules. There is a critical wave vector above which the time scale is becoming independent of the wave vector. And so these authors added a term by hand, which is proportional to the rate of change of curvature. So del 4u, del s4 is essentially curvature. And you have a local dissipative force that is proportional to the rate of change of curvature of the filament. And by adding this term purely on phenomenological basis, you can get a wave number independent time scale. So with this background, we knew that probably in cilia also the internal friction will be important because the building block itself seems to have some internal friction associated with it. There is another source of internal friction in ciliary beating that could come about simply due to the nature of motor filament interaction. So in this experiment, there is essentially a microtubule that is lying on a substrate, and you come in with a colloidal particle carrying a molecular motor, which is held in a optical trap. And then if you move this stage periodically with speed v, what you would find is that the friction force will scale non-linearly with speed v. And so what this is showing you is that there is breakage of bonds between motor domains in microtubules at high enough speeds, which is leading to the tapering of the friction force. And so, so there is some sort of friction in just the way how motor walks on this microtubule, and the mere action of walking is equivalent to attaching and detaching, and that sort of bond breaking can also cost energy. So there have been experiments which also looked at how the response of a whole cilium, inactive cilium, to shear. So here in this classic experiment, you lay down an inactive cilium on the cover slip, and then you come in with a micromanipulator with a sharp tip at one end, which is in contact with the cilia. And then you move the base of the needle with a certain displacement and measure the displacement of the tip. So what you see is that, so in terms of rheology, this is an experiment equivalent to creep. So what you see is that the displacement of the tip needle goes very smoothly to some stable value. So this long development of the displacement tells you that this is not a purely elastic filament. A purely elastic filament will have a very abrupt and quick jump in its the tip displacement. And so the gradual displacement of tip implies that cilium, I mean, even an inactive cilium has some viscoelasticity associated with it. There is a broader context, even broader context of internal friction in the context of protein folding. So people have measured refolding times of proteins as a function of the viscosity of the solvent and found out that if you extrapolate to zero viscosity, you do not get zero refolding times for low denaturation conditions. And so essentially, even in if you take essentially the what I'm trying to say is that regardless of the biopolymer you may have had at hand, as long as you have some polymer chain, you will have a source of internal friction associated with them. So I will now stop here and take questions, because then I will present to you our work.
So I do not see any question in the chat box.
Okay.
Vijay has raised his hand, Devashish.
I have a question.
Okay, yeah.
So, Prerna, when you showed one of the papers which put in some phenological
Yes.
coupling,
Yes.
what's the motivation? I mean, that's a really odd kind of a coupling. It's a mixed time-space derivative.
Right. Let me go back to that. So, I mean, see, by having the time, having the space here is what ultimately allows you to cancel off the q to the power 4.
That's correct.
So,
That's correct.
yeah, and basically because the r to the power 4 is coming because you have rate of change of curvature. So if you want to get a force kind of term while retaining a viscosity like coefficient, then you're bound to have a length scale.
No, that's not what I meant. So I meant, what is the physical motivation for including a term which is like the rate of change of the curvature?
Right. So essentially what you are saying is that there is so here you have two kinematic, I mean, in the case of microtubules you have only one kinematic degree of freedom, which is bending. So to each kinematic degree of freedom you have associated a friction term which depends on its dynamics. So it's simply that. Then there are, I mean, what is the micro origin of it is like there are slow conformational changes in the microtubule as it bends, and you can construct simple models like you could imagine having a tube of fluid within this filament which is responding to bending, and then you will get a dissipation like that.
Mm-hmm. Now, I was just wondering, maybe at that length scale, the continuum modeling is not valid. I mean, have we hit that length scale? Probably not. So this is what I don't understand.
Yeah. So this is indeed continuum modeling, and so I think it's a fair enough thing to do because you're not far from you're not far from equilibrium. Meaning, so these are like small. So I mean, microtubule is a very stiff filament to begin with, right?
Correct.
So your bending amplitudes are very small in that sense. So the deviation, if you're worried about very large bends, then no. Also, all the measurements are happening on. So the microtubule um, diameter is like 20. I mean, the diameter is 20 nanometers. The length is about. It can be as large as you want. Meaning you can polymerize it. So all the measurements are on 200 nanometer length scale, right? These are optical measurements. So in that sense, they're highly coarse grained, and this kind of modeling is probably okay.
Okay.
So there is one more question.
Yes.
Pratyush, if you can unmute yourself and ask the question.
Yeah, I wanted to. So you showed that in one of the experiments that there was a regular flow, and the motion of the two cilia in that organism were yeah, this one. So have there been experiments where they have done this with the fluids that are not very regular, and are the cilia still able to maintain the synchrony, or they don't,
So
they act weirdly?
I think what you're asking is instead of having a viscous fluid, if we have a viscoelastic fluid, is that the question?
Yeah, and also if the flow is not sort of something regular, like if it's some random flow, the
Um,
directions keep changing.
Random flow. So I'm not aware of studies which have looked at cilia in turbulent flows. If
Yeah,
see,
turbulent flow,
yeah, either you can have laminar flow or
turbulent flow. Yes.
yeah. So I am not aware of studies which have looked at chlamydomonas in turbulent flows. Yeah.
So the breaststroke motion is what I was wondering, if that is still being maintained in a turbulent flow, if the cell is able to maintain
Yeah,
near.
I'm not aware of
Okay, okay.
turbulent flow studies. Yeah.
Yes.
Most studies. So because I mean these are low Reynolds number systems, so you impose laminar flows because the the cell is at that sort of scale. But it's an
Right.
interesting question. Maybe someone can have a
Okay. Okay. Thank you.
Okay, so I don't see any further questions. So maybe Prerna, you can
yeah.
go ahead.
I will. Okay. So just reduce. So I'm going to now present our experimental work on this system. So we took the popular model organism Chlamydomonas reinhardti, and because we wanted to study isolated cilium and we wanted to be free from the control by cell body, we snipped it. So we removed the flagella from the cell, and you can do that by adding antibiotics or through mechanical means, and these cells will shed their flagella. So this flagella by itself will just be a Brownian rod-like object and move around. But if you add a source of energy, which is ATP here, then because the motors are all along the filament, they can use this source of energy and work. And so essentially a free cilium will be able to swim through the fluid. If you anchor this filament onto the surface, then you will see the characteristic ciliary beating that I've been showing you so far. So the nature of the waveform is very similar to what you would observe on live cells, and this has very simple boundary conditions which you can use while analyzing the data. So a number of studies have worked at these reactivated cilia, but and all of them have focused on measuring the waveform. So what we did was we wanted to measure simultaneously both the waveform and the flow field of this object. So by doing this, what can we get? From the waveform, we can get the elastic stresses in the system. And from the fluid or the flow field, you can get the hydrodynamic forces. The important point of the system is that the material parameters of cilia are very well known and consistent across the diverse experimental probes. By that, what I mean is what is the bending rigidity of this filament or what is the drag coefficient of this filament are well known in literature. And what we have done is we have put tracer particles in the fluid, which will help us measure the flow field. So I'll play the movie now. So you can see the filament in very high resolution, and you can also see how these beads are moving. So by tracking the motion of these beads, you can determine the flow field. So here I'm showing you the waveform, and you can discretize, you can describe this waveform using a polynomial description, Chebyshev polynomials in this case, and you can measure the flow field. So I'm showing you streamlines of the flow field for a particular conformation of the filament. We can, because we have hundreds of movies like this, we can essentially measure the flow field at each time point in the stroke. The nice thing about the reactivated system is that you can control the beat frequency of the filament as a function of the ATP concentration. So I'm adding ATP from outside, and I can control how much ATP I'm going to add. So there is a transition from no beating to oscillatory beating around 40 micromolar of ATP. And we have done our experiments close to this threshold. So the reason we stay close to this threshold is because we want to be in close to linear regime of beating, where we can model cilia as a shearable active beam with planar motion. So we can throw away most of the microscopic details, calculate stresses averaged over the cross section, and use the information from experimental data to determine the contribution of each term in equation of motion. So this is in contrast to the previous studies where you would write down an equation of motion, then try to get parameters to fit to the experimental waveform. Here what we are doing is we are getting waveform and flow field to independently assess the magnitude of each of the terms in the equation of motion before actually settling down on a given equation of motion. So the flow field is really crucial because it enables us to validate the form of the experimental drag really well. So remember, this is an extended object, and so it's not clear whether simple resistive force theory will work or not. So what we have here, I'm showing you two different conformations of the filament in power and recovery stroke with their flow fields. And we can also calculate the flow field theoretically. So essentially what we are doing here now is testing resistive force theory. So what does resistive force theory say? Resistive force theory says that the local viscous force is proportional to the local velocity of the filament. And I can measure the velocity of the filament very well because I know the waveform at high spatiotemporal resolution. So therefore you know the theoretical resistive force, and you can calculate a theoretical flow field by integrating this viscous force with the Oseen tensor, or in this case the Lorentz-Blake tensor, which accounts for the fact that there is a wall close to this filament. And essentially we can compare the theoretical flow field with the experimental flow field. So we are thinking of our filament as a line of Stokes' of strength Fv, and each of which gives you a flow field through the Oseen tensor. So this is how the theoretical flow field looks like. And you can see that there is a very good match, definitely in streamlines, but there's also a good match in magnitude. So we can measure the speed at each point on this grid and compare it with theory, and we come to a conclusion that resistive force theory works well in this case. And so now we are assured of the fact that we are using the correct form of viscous drag, and we don't need higher order corrections. So now let's look at the mechanics of the filament. And so we describe the mechanics in this tangent normal form. And as I said, we're going to model cilia as a shearable beam with diameter A. And we're going to ascribe a tangent normal at each point. And the tangent angle is theta. The key ingredient of the fact that there is shear within this filament, because the microtubules are being slid with respect to one another, that shear displacement can be connected to this observable tangent angle through a simple geometry where you see that the shear displacement is diameter times the difference in the tangent angle. So here is my waveform or the stroke of the full cycle of the beat. I can measure the tangent angle at each space point and time point and plot it as a hemograph. This tilted fingerprint texture tells you that the tangent angle is showing a traveling wave like behavior. So if you plot the tangent angle for many different arc lengths, so S is the arc length, L is the length of the filament. So you are non-dimensionalizing the arc length with the total length of the filament and time with the time period of the beat. And what you find is that you can fit a traveling wave like behavior with an average shape superposed on it. In the analysis that will follow now, we will throw away this average shape because we are interested in oscillations about the average shape. So the average shape is actually bent here. It's not a straight filament, and that is what leads to a net flow when this filament beats. So what are the force and torque balance equations here? So essentially the sum of forces acting on this fluid filament system have to add up to 0. So if you look at the cross section, there are forces that are acting on the cross section of the beam, and then there are forces that are acting on the surface of this beam. So you will sum up this in the following manner. So where basically you have gradient of the force acting on the cross section being balanced by the surface forces. You can similarly write a torque balance equation or the moment balance equation. And because we have planar motion, what you will find is that the moment balance equation will actually become a scalar equation where this capital Fn can be extracted from the force balance equation, and you can see that it will be related to the viscous force. So the viscous term is being carried here. And now what I'm going to do is I'm going to use simple constitutive equations for the moments that are acting on the cross section as well as acting on the surface by relating them to the measurable quantity tangent angle in our case. So the first obvious source of moment is the bending moment, which is related to the curvature in the following way. So the moment is linearly proportional to curvature with the bending rigidity as the coefficient. Then you can have the shear moment, which is essentially the fact that you have shear elasticity in the system, and that will be related to the tangent angle itself. Then you have moments due to the motors themselves, and with this you can essentially combine all the terms here. And so the first term is the curvature elasticity, the second term is the shear elasticity, the third term is the viscous term, and the fourth term is due to the motors or due to the molecular motors. So there are some characteristics, length scale and frequency scales, with which I'm going to non-dimensionalize this equation for a robust comparison. And now what I'm going to do is I'm going to compute all these terms other than the active moment from the experimental data and look at how they compare with each other. So this is the first term, which is the curvature elasticity, second term, which is the shear elasticity, and the third term, which is the viscous term. So the point to note here is that the scale bars here are quite different. So the curvature and shear elasticity are 2 orders of magnitude larger than the viscous force. So remember at the beginning of my talk, I had mentioned that this is a continuously driven system, which if you want to be in a steady state, you need to have a good competition or a good balance between the elastic stresses and the viscous dissipation, which in this case is not happening. And therefore I need to invoke internal viscosity for self-consistence. And I'm going to add terms by hand which will correspond to curvature dissipation and sliding dissipation. So I have two kinematical degrees of freedom here, curvature and sliding, and therefore I'm going to have friction associated with both degrees of freedom. So, so here are the additional friction terms that will go in my constitutive equation. So I'll have a curvature friction, and I can plot it by using the curvature friction coefficient of the microtubules. Similarly, I will have a shear friction coefficient whose estimate we have done from back of the envelope calculations, but whose magnitude also matches with the motor filament friction that I had shown you in the first part of my talk. And so what now you see is that now these additional friction terms are in good competition with the elastic stresses. So at least now the description is self-consistent. And what I'm going to do is I'm going to drop the fluid viscous term completely and retain only the curvature and shear friction, because the fluid viscous term is way weaker than these two terms. And so I'll get a modified equation of motion of the filament. And now I can ask whether this equation permits oscillatory solutions. So that's where the theoretical part comes in the sense that now we would like to know whether the equation of motions permit oscillatory solutions. And so I need a constitutive equation for the active moment. So I've chosen the simplest constitutive equation for the active moment, which is first order in time and related to the sliding displacement. So B1, which is a constant, characterizes the autonomous dynamics of the active moment, and B3 characterizes the feedback it gets from the sliding of the filament. So this is a coupled equation of motion. And what we did was we essentially did a linear stability analysis where we Fourier transformed the equations in space and time and looked for a transition from unstable to stable oscillations. And so in the B1, B3 phase space, indeed, there is a decaying to unstable oscillation transition. If you zoom in close to close to this area, what you would find is that there is a line of points whose limit cycle frequency will match with the beat frequency. And we have essentially identified the B1 and B3 coefficients, which will give a fundamental oscillatory mode, which will match with the experiments. So essentially here, what I'm saying is that by looking at this phase diagram of decaying in unstable oscillations, I can identify values of B1 and B3 close to the threshold or close to the transition point, which will match with the beat frequency. So that is the only constraint that I have imposed. In other words, if this constitutive equation were to model ciliary beating as observed in our experiments, then the values of B1 and B3 one should be using are these. So we have also scanned the phase space of the drag coefficients of curvature friction and the shear friction, and found that for varying values of these two, you can get a decaying to unstable oscillation transition. So now that I know that this coupled equation of motion permits oscillatory solutions, now I'm going to ask questions about what is the nature of the active stress. So I can write the tangent angle as an oscillatory form, and the active moment also as oscillatory in nature, and substitute it in this equation. So basically what you're getting is you're getting a response function kind of behavior. So how is the active moment connected to the tangent angle? It's connected through this response function, which is complex in nature, which has a in-phase part and an out-of-phase part. And so these are active elastic stresses and viscous stresses. So if K and lambda were negative, then that would mean that the system's passive spring constant friction coefficient will be renormalized by the ATP activity. So essentially it's as good as essentially changing your passive elastic parameters. But if K and lambda are positive, then the activity is working against the material response and is effectively strain softening and shear thinning. So in our case, K and lambda are actually positive and lambda is larger than K. So that means the active stresses is viscous dominated over the elastic ones in our experiments. So with this, I will conclude this part of the story that we have shown that the RFT is a valid form of hydrodynamic drag for a beating cilium. This is a direct experimental evidence of hydrodynamic drag being negligible compared to passive stresses, and internal dissipation mechanisms are essential to have a self-consistent picture of ciliary beating. So I'll stop here and take questions before I go to the next part of the talk, if there are any questions.
Prerna, I don't see any. Okay, Pratyush raised hand.
Yeah.
Pratyush, you can unmute
Yeah.
yourself.
Thank you. Actually, I wanted to ask that there was this while talking about the equation, the moment equation, you had shown these diagrams of patterns, black and white patterns. Could you explain those? What those diagrams mean? Yes, please.
Yeah, here, right? This one?
Yes.
Yeah. So
Yeah.
basically, this is how you would write. So force balance equation is easier to explain, right? Because essentially you have F plus DF, so you have to sum up these things. So basically DF plus FDS is equal to 0. And then you differentiate with respect to arc length to get del SF plus small f equal to 0.
Okay. Yeah.
So for the torque balance, you have to then think about the torques that are acting on the two sides, and that diagram is not here, so I can't easily explain. But
I'm actually talking about the graph that you've shown above in the middle.
Oh, this graph.
This pattern. Yes.
Okay. So this is space-time chemograph. So essentially what we are doing is we are plotting the tangent angle as a function of arc length. Okay. So that is this axis, and then you do it for different time points of the beat, essentially. So so which is, I mean, the same thing is plotted on the right here. So where you can see that the so what I'm trying to say is if you stand at some particular arc length on the filament and look at the tangent angle at that arc length as a function of time, it is oscillatory. And similarly, if you fix your time point and look at the tangent angle as you go for different space points, arc lengths, you will find it's oscillatory again. So
Right. Okay.
it's the same that's plotted here, essentially in a color plot.
In the following slides, the same pattern has been shown with different.
Yes.
The pattern slightly changed. What does that mean?
The pattern changes because you are looking at different derivatives of theta. So when you take a double derivative of theta with respect to arc length, this is what you get, which retains a little bit of traveling wave likeness, but of course has these distortions because you've taken a double derivative with respect to arc length. So whereas delta theta is just the same, it's the same plot as before because you're looking at tangent angle itself. What you can see from the third plot is that now instead of a tilted fingerprint, you have more like a straight thing. And that tells you it's not a traveling wave like in nature, it's more like standing waves. But more than that, there is no physical, in that sense, no more physical interpretation. These are various, so the viscous term is some integro differential term. And so it has all kinds of derivatives of theta and time mixed in. So not easy to, in that sense, motivate. Yeah.
Okay, thank you.
So next, Samarth Sabu has a question. Can you?
Yes, thank you. So this is from the first part. You had shown that about synchronization of the cilia.
Yes.
So I was curious to know, like you had mentioned about the bridge or connection in between, which intracellularly kind of makes the synchronization happen.
Yes.
So I want to know, like, have there been experiments in which that has been cut or removed, and they?
So there have been experiments. Basically, you prepare mutants where you can, disconnect these distal fibers.
Okay.
So there are mutants of Chlamydia monas where this distal fiber is compromised, and there you find that synchronization is not present or present weakly and things like that. So there has been a whole range of studies which look at, fiddling around with this distal fiber. Yeah.
Okay, thank you.
So Benjamin, you have a question. Can you unmute yourself?
Yeah, if I may. So, Sharma, in your model, you have the elastic stresses of the band cilium, which of course requires knowledge of the elastic modulus.
Yes.
I'm just curious how you estimate this. I only know the old literature, and there's a whole range over one order of magnitude of different bending elasticities, whether the motors are bound or not.
Yes.
So what is your answer to the value of the bending modulus of cilia?
Yeah, so we have used the highest bending modulus available in the literature for cilia.
Okay.
So because we were comparing, we were aware of this discrepancy, and we wanted to be sure that no matter which value of bending rigidity we take, it was still way larger than viscous stresses. Yes.
And do you actually require these elastic deformations? So if in your model you showed the lambda, the viscous internal viscous contribution is larger than the internal elastic contribution. If you would set the K, the internal elastic contribution, to 0, would you still get oscillations?
So I'm sorry, I didn't get which coefficient you would like to put.
Okay, if you would set the bending modulus to 0, would you still get oscillations?
A priori, I don't know the answer, but we never put bending modulus to 0 because that didn't seem like a physical thing to do. But I mean, so it's equivalent to putting del S2 delta theta term 0, right?
Mm-hmm.
Equivalent to putting dropping of this term. So I'm not sure whether we will get oscillations or not. Yeah.
Probably not. So it's okay. So it's small, but still necessary.
Yes, definitely.
Okay.
Yeah, I mean, it's elastic. It is a crucial term, I think, because you have a very stiff filament that you are bending, and so those stresses must be accounted for one way or the other.
Thank you.
Thank you.
So, Professor Vidyanand, Nanjundiah, you have a question?
Yeah, I'm sorry, this is a bit of a delayed question. It refers to the start of your talk. You seem to have treated the flagellar and ciliary movement as being essentially the same. Is that reasonable?
So the term flagella and cilia were used interchangeably. I was referring to eukaryotic cilia. And eukaryotic cilia is basically motor filament assembly, molecular motors and microtubule assembly. And then there is prokaryotic flagella, which is present on bacteria. where you basically have a polymer-like system only made of flagellin molecules, which is driven by a rotary motor that is present inside the bacterial body. So that flagella and this flagella are totally different microscopically as well as in terms of the beat.
I understand. My question was, in the beginning, you seemed to use the two words interchangeably.
Yes,
And I thought that was because in terms of the dynamical analysis you're giving, you could refer to either of them. Is that correct?
Yes, I was referring to them interchangeably. And within the context of eukaryotes, cilia and flagella mean the same thing. So you just have to be aware that there is this other class of flagella in prokaryotes which behave very differently. And they function, I mean, their microscopic mechanism is also different. So I'm not referring to those flagella.
Yeah, sorry to push this, but what you call eukaryotic flagella, do they ever work in bundles?
No. So this
I thought that was a basic difference.
Yeah. So the bundling of flagella happens in prokaryotic flagella. I mean, the classic bundling where multiple flagella will come together.
Right. Yeah, and that doesn't have any effect on the dynamics as you are analyzing it?
So I'm saying that our analysis is not applicable to the flagella that bundle and, work through this rotary motor. Yeah.
Thank you.
So, Prerna, I also had a question, quick question. So this about the internal frictions, internal dissipation due to bending and shear.
Yes.
So do you think they are coming from the same microscopic process? Could be just due to the cross-linking motors, or maybe some passive cross-linkers, I don't know.
Yeah, so the thing is that the two, I mean, we have sort of classified the internal friction as the curvature friction and the shear friction, mainly because they depend on these variables differently. Like one depends on the rate of change of shear strain or shear displacement, and the other depends on rate of change of curvature. But also the curvature friction, I think, is more like coming out. It's a passive contribution. It could be due to the Nexin links, or it could be due to the microtubules themselves. And the shear friction, we expect it could be due to the motors. We are not very sure. We have again used back of the envelope estimates of protein friction within the cilia. So the Nexin links that I'm talking about can bind, unbind, and so looking at that, we have estimated the shear friction term. Whereas the curvature friction term we have used from the microtubule experiments. So the numbers that have gone in are from these two different contributions. Yeah.
And how do you define? How do you identify, differentiate the active torques or active forces here?
So these are not active torque. I mean, I'm sorry. So
this term I thought is due to the activity.
Yeah, you mean del T MA plus B1 MA, right?
Yeah. Right.
Yeah. So sorry, I got confused. I thought you were referring to active dissipation. So that's what I was saying, that we haven't modeled any.
Yeah, both. So I was thinking about both active dissipation or maybe passive crosslinkers. Are they present there?
Yes, passive crosslinkers are present aplenty in this organelle. And so these shear friction and curvature friction are related, in our description, are related to passive crosslinks. And this equation for the active moment, del T MA plus V1 MA equal to B3 delta theta, it has been chosen by hand, in the sense it is the simplest constitutive equation you can come up with for an active moment. There are you could have even more complex constitutive equations. For example, you could have something which depends on rate of change of tangent angle, or you could have non-linear, you could have higher dependencies on the tangent angle and so on. But we didn't do that because this was more like a
linear response.
Linear analysis, and where you want to see whether simplest models can give you oscillations or not. That's all.
Sure. Okay, thanks. So I don't see any further questions, so maybe you can go ahead with the rest of the talk.
Yeah. So, all right. So now we will switch gears and talk about phototaxis. And here the friction is not the friction that I've been talking about in terms of fluid friction or internal friction. It will be slightly different in flavor, but the ideas are actually relevant. So Clemmie usually walks under dark as in a persistent random walk manner. So what you can see is that the cells are moving randomly within this chamber. So you're looking at a highly zoomed out view. You're looking at multiple cells, and you can see that they move randomly. And you can measure what is called the rotational diffusion coefficient of these cells. So I'm plotting the mean squared angular displacement versus time for these cells for many different cell concentrations. And the slope of this graph, or the long time slope of this graph, gives you the rotational diffusion coefficient. And this rotational diffusion coefficient is independent of cell concentration. So essentially, I've characterized the persistent random walk by measuring its rotational diffusion coefficient. If you place these cells in presence of a blue light source, they will run towards the light source. And if you watch this movie a little bit more carefully, you will see that some of the cells are actually running in the wrong direction. And so, and the system is in steady state. So we have kept these cells in blue light long enough to see this kind of behavior. So here I am plotting the XY trajectories of these cells. So each color represents a single cell trajectory, and all trajectories have been offset to start at 0, 0. So essentially 0, 0, that's where all these trajectories are starting. And so you can see that a large fraction is running towards a light source, but then there is a small fraction that is running in the wrong direction. So if you plot the probability density as a function of polar angle for these trajectories as a function of light intensity, you will see that for pitch dark, the probability density is flat. That means the cells have equal probability to run in any direction. And then as you increase the light intensity, you find that you develop a peak which is in the direction of the light source, and the peak height goes up as the light intensity goes up. So this is the simple stimulus response behavior. So this behavior we expect that if you increase the light intensity, a larger number of cells will run in the right direction. What was interesting in our experiments was that we found a cell concentration dependence. So here I am plotting XY trajectories as a function of different cell concentrations. So what you see is that at low concentrations, the cells are running towards the light source and some are running in the wrong direction. As you increase the cell concentration, there is a larger fraction that is running in the wrong direction. And then as you increase further, this fraction again comes down. So there is some kind of re-entrant transition. So if you plot the probability density offsetted for the direction of the light source, you find that the peak height goes down as you increase the concentration and then goes back up again. You can also measure what is called the phototaxis efficiency, which is the fraction of cells that are running in the direction of the light source plus minus 10 degrees. as a function of cell concentration for two different light intensities. So for low intensities, you can see there is a very clear dependence on cell concentration that is re-entrant in nature. That means it has a well-defined minima and a pickup. And then for higher light intensities, this dependence becomes weaker, as you would expect once again. Because if you are at very high light intensities, to begin with itself, you have very high phototaxis efficiency. So the markup that you will get will become lower. So we used a very simple model to describe what is going on here. And I will also show some experiments which will highlight what is the origin of this behavior. So we are essentially looking at a model of a self-propelled particle in presence of an external potential, which tends to orient the cells along the light direction. So this is a simple dynamical equations for the position and angle of the cells. And then there are some noise terms, as the thermal noise terms. So if you write down the Fokker-Planck equation for the angular probability density, it's a diffusion equation with an additional term due to the external potential that we have imposed. And the solution to this equation is a von Mises distribution, which looks like a Gaussian in appearance and has the following form. So from here we can, if we fit this expression to our experimental data, we can extract the strength of the potential. And so here the points are the experimental data and lines are the fit to the von Mises distribution. And you can see that we can extract this strength of the potential that has a behavior very similar to the experimentally measured phototaxis efficiency that we had measured. And so the model is a good description of what is going on in some ways. But of course, we don't know why the potential should depend on the cell concentration to begin with. And so that origin, we can explain above the critical concentration. So essentially what we were asking is that why does the phototaxis efficiency go up as you go above the critical concentration? And then our initial hypothesis was maybe the cells are secreting something which is helping them help each other in terms of finding the light. But exchanging media and things like that, even careful experiments did not point to a chemical signaling pathway. And so we sat down to look at more physical pathways, such as, do the cells slow down? Does something happen to the speed of the cells? And what we found was that yes, the speed of the cell was decreasing as you increase the cell concentration above the critical concentration. And so above critical concentration, the cell speed is coming down. And the same is happening whether the cells are in blue light or they are in pitch dark. Their speed is goes down as you increase the cell concentration. And that is intuitive in the sense under crowded conditions, the cell speed is bound to come down. So what does this have any repercussion on how they detect light? So far I haven't spoken to you about how they detect light. So these cells have a single single eye spot, which has multiple photoreceptors within it. And this cell swims in a helical trajectory while the cell body undergoes rotation about its own axis. So essentially what is happening is that the antenna is able to scan the space in 3 dimensions and determine where the light is incident. So when you can visualize this rotation of the eye spot by using polarization microscopy, where the eye spot appears as a bright object and the cell body remains dark. So I'll play this movie and you can see that the eye spot will rotate, going from one side of the cell to the other side of the cell as the cell moves in the field of view. So what that tells us is that if you slow down a cell, its rotational frequency will also slow down. In fact, we can measure the rotation frequency as a function of cell concentration, and we do find a decrease in the rotation frequency. And so how will the rotation rate going down help? The rotation rate will help because if the cell body is rotating slowly, it will enable the cell to collect more photons per unit time and therefore detect the light direction more accurately. That is our working hypothesis, that perhaps this is the reason slowing down the cells will enable them to detect the light direction better and therefore have a higher phototaxis efficiency. So we independently also tested this assumption by putting cells in more viscous media by adding a polymer called methyl cellulose, where we could see, okay, we are able to decrease the speed as much as we were able to decrease the speed by crowding with other cells. And so now we can plot the phototaxis efficiency as a function of mean speed for with methyl cellulose and without methyl cellulose for the same concentration. And so that tells you, so this kind of curve tells you that our hypothesis is probably correct, that the mechanism of enhanced efficiency is the decrease in the linear speed of the cells as the concentration increases. And so we have given a simple physical and phenomenological mechanism that gives you, that shows the observed complexity of the collective phototaxis. So I will stop here, and if there are any questions, I can take them.
Okay, thank you, Prerna. Nice talk. So are there questions?
Yeah, I have.
Yeah, raised hand. Please ask.
So Prerna, the modeling with the ABP-like
Yes.
model that you showed,
Yes.
and you put in a potential, and you put in a potential only in the angle theta.
Yes, yes.
So, I mean, I thought you saw the trajectories in the real space, right? In the physical space.
Yes, yes.
But now you're putting an angle theta.
Yes.
So, but the theta is some internal coordinate in the particle.
Yeah, I mean, we are visualizing the trajectories in real space, but we are interested in the angle itself, in the sense that when we, so let me, so for example,
So, sorry, just remind me. The theta is the direction of the propagation with respect to some fixed axis. It's fixed.
Yes, yes. So theta is the direction of the motion of the cell. And so when we compute the phototaxis efficiency, we are looking at fraction of cells that are within few degrees of the light source. Right. And so I mean, the microscopic picture is that the cell is running in a certain direction, and then when you turn on the light, it has to reorient itself in the light direction. And so the force is on the direction of motion rather than on the speed.
Right. No. So no, sorry. What I meant was, is theta measured with respect to the light source direction or some fixed axis?
It's measured with this. So the source is.
It shouldn't matter. I mean, if you
shouldn't
have
matter.
the fixed,
basically, you fix one axis, whether it is the light source axis or you fix, and then you measure.
Fine.
Measure all angles relative to that axis. Yeah.
Right. No, but the sine theta potential that you put, the theta is the angle with respect to the light source, is it? I don't know how you...
So when you put a gamma sine theta, that what that is telling you is it's asking the cell to align along 0 degrees.
Which is going towards the light source?
Yeah, so light source is placed at 0 degrees. That is the convention. Yeah.
Right. And I missed the axis. You had shown the plot of the potential as well.
Right. No, I didn't show the plot of the potential.
The fit is somewhere there.
Yeah. So here actually all the angles are offsetted for better visualization.
Okay. So same thing. It's not a multiple. Okay.
Fine. I just offsetted the central position so that you can see that the peak height goes down and then goes back.
Sure. Okay. Thanks.
So Benjamin, you have your question. Can you unmute you.
Yeah, if I may. So you put forward the interesting hypothesis that the increased phototaxis efficiency at higher cell density could be due to the cells collecting more photons.
Yes, yes.
I wonder, could one test this hypothesis by increasing the total light intensity?
Yes.
And thereby also changing the expected photon count?
Yes. Yeah, so we did take data at 2 different intensities, right? So, which are different by 1 order of magnitude in number. And so what we see is that for higher intensities, there is a much weaker dependence on cell concentration than for lower.
Okay.
Yes.
That's okay. That supports your
Yes. Yes.
your hypothesis.
Yes. That is in
I was. I'm curious. It's probably a complex system, and I remember that Kishimura showed previously that the efficiency of Chlamydomonas phototaxis depends very much on the frequency of light stimulation, and that there has to be a match between their own frequency of cell rotation and the light stimulus. And I'm wondering if they swim slower, they sort of shouldn't they change the frequency at which they receive the oscillatory signal, and maybe then they are moving closer to the optimum in their frequency response set by their intracellular signaling system.
I think you're referring to experiments where they had periodic flashes of light, right?
Yes.
Yeah, but in our experiments, we have a constant source of light. So the light is always on in that sense. So this, in that sense, now I think what you're asking is the rotational frequency that we have measured, how will it connect if I were to have flashes of light? Is that the question?
No, the question was rather in mathematical models of helical clinotaxis like Chlamydomonas uses. It matters what is the frequency of the oscillatory stimulus. And I'm wondering if the cells swim slower at higher cell densities, they will receive an oscillatory light signal at a different frequency, which could actually elicit a stronger reorientation response.
Yeah, I think what you're saying is that because the cell is rotating itself, it is seeing a light dark cycle as it rotates, right? Like,
yeah.
The fact that if I rotate slowly, I have a longer dark period as well as a longer light period. So it should not matter in that sense.
Yeah. Well, then it depends on the details of the signaling system, how the cell responds.
Yeah. That's something which I don't have a good handle on in that sense, yes.
Was just curious.
Yeah. I'm in that sense, our model is not so, it is not operating at those scales in that sense. But it's a good question. It's something we wondered ourselves. But the simplistic picture is at least self-consistent and convincing enough, I believe, that we can ascribe density-dependent motility as one of the important factors, if not the only. Yeah.
So there is one more question from Samarth Sabu. Samarth, can you unmute yourself and ask your question?
Thank you for the talk. In the graph that is shown in this slide,
Yes.
So initially we see a decrease in efficiency, right? So what could be the reason for that?
Yeah. So we don't know we don't have a very convincing experimental proof as to why it should decrease, but it could be possibly self-shading, that initially when you go from few cells kind of concentration, so a very low concentration basically, which corresponds to few cell per field of view, to something slightly more, then the cells can shade light from one another, meaning the capture efficiency of other cells goes down due to the presence of their neighbors. And that self-shading then has to be overpowered by this density-dependent motility, that even though you have some kind of self-shading going on, the fact that you move slowly, you are able to still detect the light much better. But we don't have any experimental proof that self-shading is at play at below critical concentration.
Okay.
Thanks.
Thank you so much.
So Prerna, I had a quick question.
Yes.
So these cells, even in the absence of light, they are self-propelled, right? They're propelling you.
Yes, yes.
Showed in some. So my question is that in that system, even in the absence of light, if you change concentration, do you have this measured of what is, let's say, the orientational correlation of these cells propelling in different directions between cells?
Yeah, so the orientation, yes. So that was the first graph I had shown. The orientation or the rotational diffusion coefficient stays the same.
Yeah, so but this rotational diffusion coefficient is for a single guy. I'm asking a different question between cells.
Sorry, it's not for a single guy in the sense it's averaged over many cells.
True. But this is this gives you the delta theta here gives you the persistence of average cell.
Yes.
I am asking a question that if I try to look at orientation of one cell with respect to the orientation of another in the sort of in a flocking sense, in the Vijaykumar kind of flocking sense.
Yes.
So I was wondering, do they, because you are changing the densities by several orders of magnitude.
Yes.
So is there a possibility that the cells gets into a flock? So even in absence of light, they will get to form flocks?
Yeah, we tested it and actually it was flat. The probability density, you can tell from the probability density plot. So black is the dark line, or when there is no light. And then for all concentrations that we had used, it was the same. So we had excluded that possibility.
But isn't that again measurement? So isn't that again measurements over individual cells and then taking the data altogether? What I am asking is the correlation between two cells.
Yeah, so we haven't measured the correlation between two cells, but what I was referring to, if it were flocking in a more macroscopic manner, then it would have reflected in the probability density, because I'm looking at XY space trajectories, right? So
but if you have long enough trajectories, then the flock can rotate in all possible directions.
Yes, that's a possibility. We haven't measured it, but the microscopically we know clammy is a puller kind of swimmer, and pullers usually do not have alignment interactions. If you were to have a pusher microorganism like E. coli, then alignment interactions and swarming are much more likely.
Okay.
So in that sense, we were not we did not test it, but we can definitely test it with your suggestion. Yeah.
Okay. So I don't see any more questions here.
Yeah.
So maybe then we can, all of us can thank Prerna for a fantastic talk, and let's come back again at 4:30 for the next talk. By Benjamin, you have a question or no?
No, I was trying to clap
This claps. Okay.
Thank you, Prerna.
Okay.
We'll come back in Mana.
Bye.
Okay.